wait for one more minute. Students are joining now, right now. Okay, so let's see. Let me give you 1 minute. All in, I feel. Yeah, yeah. Very good evening. Good evening, guys. Good evening. I hope that's it for today's class. 17 people. Okay. Um, I think I'm good to go from here. Anyways, I'll be revising from the past class, and we'll see if your friends wait so that they don't miss anything, right? So let's go to our slide and get back here. So we had a nice introduction to molecular basis of inheritance, as I was saying, and I was also talking about the history of how this thing started. Okay, and among that, when we talk about the scientific era, I was also mentioning about this nucleic acid and how the nucleic acid was discovered by Frederick Michner, and while he isolated these pus cells. So he isolated its pus cells, and I told that he named it as nuclein. And after that, the acidic nature of the nuclein was again discovered by Altman, and then he gave the name called as nucleic acids. So in our biology, we also discussed that we have two types of nucleic acids. One is deoxyribonucleic acid, and the other one is ribonucleic acid. Correct. So when we talk about the DNA or the deoxyribonucleic acid, and I also mentioned so many factors that comes with DNA, right? So I told it's a long polymer of deoxyribonucleotides, and its length depends from one organism to another organism. Like the eukaryotes, like human beings, have a different length, right? Whereas the bacteria has a different length, and bacteriophage has a different length. It depends upon the number of nucleotides DNA is made up of. So that is what defines the length of the DNA. That's what we talked about, right? So here is where we had to continue, and I was telling you to make DNA, or to for a DNA to be present, it is made up of three important ingredients, right? Like that of our coffee. We took coffee as an example. So if I asked you to make coffee, I would bring milk, sugar, and you would bring the decoction, or the black thing, or the brew of the coffee, and you mix it together at the right temperature. You get the best coffee, right? So similarly, when we talk about the composition of the DNA, and the DNA is made up of three important components, as I mentioned. What are those, students? It's given in your slide. You just have to read it. Okay. So first one is deoxyribose. Okay, for the cursor, deoxyribose sugar, and then we have a phosphate group, and then we have a nitrogenous base. So what is a deoxyribose sugar? So usually there is a question that comes up in your practical examinations, or in CET, or common interest test, or the state level examination. So they come up with this question very frequently: what type of sugar is present in the DNA? So first of all, we are talking about a ribose sugar. Okay, sorry, we are talking about a pentose sugar. Now, what is a pentose sugar? Now, if you compare your normal sugar or the glucose, the formula for the glucose, which you already know, is C6H12O6, isn't it? So in the C6H12O6, the number of carbon in this C6H12O6 is 6. So that is why we call our glucose as a hexose sugar. Whereas when we talk about the sugar that is present in our DNA, it is made up of 5 carbon atoms. So that is why it is a pentose sugar. And what type of pentose sugar? It is a deoxyribose sugar. Understood? So this deoxyribose sugar, what we have is having 5 carbon atoms. And the carbon atoms are very, very crucial for us to understand how they are linked with each other. Okay, and the next one is the phosphate group. So usually the donor group for this phosphate characteristic of our DNA comes from phosphoric acid, that is H3PO4. All right. So we'll talk about more details about this phosphate group. And next we have is our nitrogenous base. So when we see our nitrogenous bases, they are very, very important. So in our nitrogenous bases, in our DNA we have 4, and again in our RNA we have 4. So in general, in nature, we have 5 types of nitrogenous bases that is present in our nucleic acids. So what are those? One is adenine, guanine, cytosine, thymine, and uracil. Okay, we have 5 of them. So if you take our DNA has ATCG, that is adenine, thymine, cytosine, and guanine. Whereas our uracil is not present in DNA. And similarly, if you take RNA has adenine, guanine, uracil, and cytosine. There is no thymine. You understand my point? So in DNA we have thymine, and in RNA we have uracil. And rest 3, that is ACG, is common in our both DNA as well as RNA. All right. So when we talk about our DNA, right? Our DNA, as I told you, one is the phosphate group. I told you is H3PO4, and then we have our nitrogenous bases, and then we have our deoxyribose sugar. Now our focus should be on understanding our sugars. So our sugars comes up in 2 different forms. One we call it as purines, other one we call it as pyrimidines or pyrimidines. So what is the reason we call them as purines and pyrimidines? Okay, so it's very easy, students. So let us take a look at this particular structure. Give me a second. Okay, you take guanine. Where is this? Okay, you take hold on. Yeah, you take guanine, and then you take adenine, and then you compare with these 3 structures that are to your right. Right, we have these 3 structures that are to our right. So you just compare with them. What do you see here? Structurally, what can you see? You can see that the 2 structures to our left is made up of 2 different types of rings, and the structure to our right is made up of only 1 ring. Okay, or 1 type of ring and only 1 ring, right? So why did we have to give them names? It's very simple. So if you see our purines and if you see our pyrimidines, so how do we recognize them? First, we'll talk about pyrimidines so that it will help you understand purines. Okay, so pyrimidines, what you can see here is, see, 1, okay, give me 1, 2, like that. We have 1, 2, 3, 4, 5, 6. There are 6 atoms in this pyrimidine ring. So among the 6 atoms, what you can see is there are 4 nitrogen. Okay, forgive me for my handwriting. Why? Because the pen tab has a problem. Okay, therefore there are 4 nitrogen and there are 2 carbon atoms in this ring. If you can see clearly, okay. If you can see the pyrimidine ring, you can see that there are 4 nitrogen and 2 carbon atoms. Okay, just see in all of these rings there are. Any ring which is composed of these atoms are called as a pyrimidine ring. Understood? So that is why they are called as pyrimidines. As simple as that. Okay. So whereas if we talk about our what do you tell? Our purines, what we can see is there are 2 different types of rings. One is a pyrimidine ring, and there is another ring which is made up of 5 carbon atoms. So, 5 atoms. So what is this ring? So this ring is called as a pyrimidine ring. I'll just annotate it as P. Whereas what about this ring, the green small ring? So this is called as, I'll just try to write it, imidazole. Okay, it's so hard. Okay, imidazole. 1 second. Okay, imidazole. All right. Imidazole ring. Sorry, students. Okay, imidazole ring. So what do we have in this imidazole ring? We have 3 nitrogen. We have 3 nitrogen, and then we have 2 carbon atom. You see, correct? So if we compare our purine and our imidazole ring, one ring has 4 nitrogen and 2 carbon, other ring has 3 nitrogen and 2 carbon. Now this is a heterocyclic structure. because there are two different rings participating to form one compound. So that is why we call our purines as heterocyclic structure, because it is made up of two rings. One is called as a purine ring, other one is called as an imidazole ring, and the examples for our purine is guanine and adenine. Okay, so is it clear for you? Yes, are you guys able to understand? So, if you have doubt, you just put a message so that, okay, you can yeah, I can just look into it. Let's go further. Okay, so next one, after we study our purines and pyrimidines, and then we started studying about our uh, deoxyribose sugar, and we also tried to understand there is another component, and that component is called as a phosphate group. Now let us try to create a chain of DNA, okay, or we'll try to make DNA. It's very, very simple. It starts with a nitrogenous base getting attached to our deoxyribose sugar. So when we talk about the attachment of our nitrogenous base to our sugar group, now many of the students are smart enough to remember this very efficiently without any problem. They don't have any problem, or all right. Whereas some students struggle to remember the numbers of the carbon atom and where they are attached. So before going to all this explanation, let us make it as simple as possible. So we have a pentose sugar. So we have a pentose sugar, and this pentose sugar has 5 carbon atoms. So this is 1, 2, 3, 4, and here we have the 5. I will write it as 5 here. Okay. And I have here 1. Okay. So this is our first carbon atom. Now, sir, I might get confused between these numbers and the carbon atoms. What is the easiest way I can remember? Just remember, whenever you talk about DNA, we will talk about 5 prime end and 3 prime end. Remember? Correct? So whenever we talk about 5 prime end and the 3 prime end, it is very, very easy that, oh, 5 prime phosphate end, 3 prime OH group. Okay. So we keep talking about this even in future classes. Always remember that the 5 prime is designated for the phosphate end. Okay, we'll just write it as P. Always the 5 prime end goes with what? The phosphate group. And the first carbon atom, okay, now this is where we talk about. To this first carbon atom of our pentose sugar, okay, a nitrogenous base gets attached. Either it is adenine, guanine, cytosine, or thymine. Okay, so let me repeat. To the pentose sugar, to the first carbon atom of the pentose sugar, our nitrogenous base comes and gets attached through N glycosidic linkage. What is the type of bond we are talking about? We are talking about linkage. N for nitrogen, G for glycosidic because it is a sugar group, and that is what the type of bond, that is what the type of covalent bond that we have. Okay, N glycosidic linkage happens between the first carbon atom and then our nitrogenous base. Now what happens? The chemical composition changes, right? So what do we call this compound where we have a nitrogen base and a sugar group attached? So this is called as deoxyadenosine. Similarly, we have deoxygynosine, deoxycytidine, and then we have deoxythymidine as our nucleosides. Okay, so S for side. Okay, nucleoside. So first thing what happens, a sugar group, you have a nitrogen base, and then they come on attached. What position they attach? The first carbon atom of your pentose sugar they attach. Okay, through what linkage? N glycosidic linkage. So after attachment, what happens? They become nucleoside. So what are the names of the nucleoside? Deoxyadenosine, deoxyguanosine, deoxycytidine, and deoxythymidine are the names for our nucleosides. All right. So let's see what happens here. And the next step is the formation of a nucleotide. So what happens here? Once we have our our sugar group, this is our sugar group, right? So this is our sugar group. Okay, and this is our first carbon atom. You can clearly see here. To our first carbon atom, what has happened? You can see that there is a nitrogen base attached to our first carbon atom to form a nucleoside. To this nucleoside, what happens? A phosphate group gets attached through the fifth carbon atom. Remember this, students, okay? To the fifth carbon atom, what happens? A phosphate groups comes and gets attached. So once the phosphate groups gets attached, okay, through what linkage? Through phosphoester linkage. Remember this, okay? Through phosphoester linkage, now the phosphate groups comes and attaches. Now this combination of these three compounds will result in a nucleotide. Again, the chemical composition changes. Now we have our uh, our entire three components combining to form nucleotides, and their chemical name would be deoxyadenosine monophosphate. Why monophosphate? There is only one phosphor group that is present. That is why we call it as monophosphate. Next one, you see deoxygynosine monophosphate, deoxycytidine monophosphate, and then we have deoxythymidine monophosphate. So these are our nucleotides. All right. So we have our nucleotides. This is one monomer. That means if I ask you to bring a chain that is made up of the beads of pearls, okay? So let's say that one ball or one beads is your monomer, and a system of these will make it a polymer, right? So we just have one of this. This one component is what we are talking about, this, correct? So this is what is represented here, okay? So this is about nucleotides. Understood? Now, what do we have here? Now we have to talk about how a polynucleotide strand is constructed. So let me go back a little bit. If you just ask anybody who is a general biology student who has just passed 10th class, if you see them, even though they don't know the structure of DNA, you would write something like this, correct? We are all fascinated by this structure. So somehow, even in our childhood, we will remember writing this structure. We don't know what it is, but somehow we were attracted to this structure because many of the science books were curated with these pictures, right? So anyways, so when we look at this, what we can see is it is made up of two lines. That means each line represents one strand or one chain, we can tell. So when we talk about a polynucleotide strand, we are talking about one of these, not both of these. We are talking about one of this. How did one chain get manufactured? So if we understand how did one chain get manufactured, then we can go up to how these two chains come together, right? So we are talking about the making of the polynucleotide chain. And here what happens is you have a chain of polynucleotides. So that means we have one nucleotide like this, right? We had one nucleotide like this, and then we have the other nucleotide coming up. So let's see what is happening here. If you see the polynucleotide chain, our sugar group is made up of 5 carbon atoms. So that was simple. So when we talk about the 5 carbon atoms of the sugar group, so I told you 1, 2, 3, 4, 5 are the carbon atoms, and we know that the first carbon atom is given to our nitrogen group, and our fifth carbon atom is given to our phosphate group, and then what is the remaining? And the remaining one, what we have is our third carbon atom, okay, which is very, very important. So let's see here. Now I'll just try to make space here, okay? So we have one, okay, let me try to make this, okay? We have one sugar group, and then below it we have another sugar group of a different nucleotide, okay? This is so, yeah, anyways. So we have our fifth carbon atom here, right, students? We have our fifth carbon atom, okay, number 5. And then here we have our third carbon atom. Here we have our third carbon atom, okay? Again, here we have our fifth carbon atom, isn't it? So now what happens is, okay, now the fifth carbon atom's phosphate group links with the third carbon atom's OH group. So we have a phosphate group, okay, we have a phosphate group, and then we have a OH group, we have a phosphate group, and then we have a OH group coming together. So when they come together, we have a bond that is called as phosphodiester bond. So when it makes a phosphodiester bond, now usually what happens is the water molecule is removed between this OH and the phosphate group, Now it will make a strong bond between the two nucleotides, thereby creating a chain. So one nucleotide connects with another nucleotide from the 5 prime carbon atom of one, and the third carbon atom of the another nucleotide comes together and they form this bond. So once they form this bond, phosphodiester bond between them, now a chain is formed. Okay, did you understand how the polynucleotide chain is being made? Okay, is it clear, students? Okay, are you able to get it? Okay, let me know faster. Yes, give me a thumbs up. It's emojis are just before you. Okay, you make use of this emojis. Okay. Yes, perfect. Okay. Others, if you have any doubts, always remember you can just ask me. Okay, at any point. So, chalo. So let's go. So now we have a polynucleotide strand. So let's get back a little bit. What did we understand? To form a nucleotide, what carbons are involved? First carbon and the fifth carbon, right? So to form a polynucleotide chain, what carbons are involved? Third carbon and the fifth carbon atom, right? So our major carbon atoms that we have to remember is carbon atom 1, carbon atom 3, carbon atom 5. Okay, I already gave you a clue. So what is that clue? I told you that to remember this, first you have to give a fixed position to one carbon atom. What is that? Fifth carbon atom. How can I remember fifth carbon atom? It's very simple, because while we study, we talk about the 5 prime end. So when we talk about 5 prime end, you tell that there is a free phosphate group in the 5 prime end. Yes, the free phosphate group in the 5 prime end is because of the presence of your phosphate group. Similarly, then you have your 3 prime end. 3 prime end, you tell that there is a free OH group in the 3 prime end. Now you have your OH group in the 3 prime end. As simple as that. Now you have your first carbon atom. Now you already know what takes its place. Okay, that is the nitrogenous base. Okay. Now let's go to the structure of our DNA. Now, the DNA structure, studying the DNA structure, it became a revolutionary discovery for the mankind. Why? Because this set a tone to understand how the DNA functions, how does it replicate, and how the DNA carries this vital information. See, it's like a stepping stone, okay, to understand more and more details about the molecular biology of our DNA. Now, the famous scientists who are credited for the discovery of the DNA structure are Watson and Crick. Okay, and guess what? Even now, our Watson is alive. Okay, and he had come to India several times. Okay, so it's very nice to see heroes alive. Anyways, so these people, they did their work in 1953, and it got recognized, and they won the Nobel Prize in 1962. So according to this Watson and Crick, they proposed this, okay, double helical structure of DNA, or which you call it as Watson and Crick's double helical structure of DNA. They proposed. Now, before we talk about Watson and Crick, now we have one more person that is very, very important for us here. So if you look at their work, basically, they had to study the X-ray crystallography studies that was already X-ray crystallography studies were already done by this woman. Okay, so she did the X-ray crystallography work, and this is your homework. Just try to find this person who conducted the X-ray crystallography, and try to remember her name. It's very, very important. After she could not understand the what kind of structure she is studying and how to give the proper explanation for that structure, and these studies were taken up by Watson and Crick, and Watson and Crick conducted their observations on this, and they constructed this ball and stick model, and then they came to a conclusion for what we study the double helical structure of our DNA. Okay, so anyways, so according to Watson and Crick, now they give the explanation for how this DNA is constructed. So the type of DNA that we are studying under Watson and Crick model is bDNA. Okay, remember this. We are talking about bDNA. So why bDNA? There is a reason. As we compare with other types of DNA models, then we understand why we call this specifically as bDNA. So in majority of our living organisms, whatever the model that we are explaining now is what is present. So that means all 99.99% of our living organisms is composed of bDNA only. So that is why we try to emphasize this particular structure as much as possible. Okay. Now we know that our DNA is a double helical structure. What is the meaning of double helical structure? It is made up of two polynucleotide chain and they are twisted together. So that is what explains the double helical structure of our DNA. Now, this double helical structure of the DNA is made up of two chains, and either of the chains, okay, let's say this is chain 1 and this is chain 2. Okay, so if you observe these two chains, they are not parallel, but they are anti-parallel, and they also show some polarity. So what is the meaning of anti-parallel? So if I take two trains and if I name the train's engine as P and the tail of the engine as the OH, okay, I'll name it like this, OH. If one train runs like this in one direction and the other train runs in the opposite direction, so this is called as anti-parallel nature of the DNA. And they also show polarity, I mentioned. So what is the polarity of our DNA? Our DNA's polarity can be explained by this. If I call this train engine or the phosphate end as the 5' end and then OH end as the 3' end, so that is what is the polarity. Usually what happens is, whenever we study the structure of the DNA, people just talk about the anti-parallel nature of the DNA, and we call one end as the 5' end and the other end as the 3' end. And we also explain, if one chain runs from 5' to 3' and the other chain goes from 3' to 5', right? So there are opposite. So that's what we explain. But we neglect the chemistry behind it. The chemistry is very, very simple. So if I give you a double helical structure of DNA, and if I ask you to recognize which is a 5' end and which is a 3' end, we are talking about molecules and atoms. So when we talk about molecules and atoms, basically you would not be able to recognize by your bare eyes. So you need a chemical analysis for understanding which is the 5' end and which is the 3' end. And that is very, very simple. If you observe the 5' end, there is a free phosphate group. Okay, a phosphate group projects like a head. Understood? Whereas if you see the same chain and at the end of the chain, you see a free OH group. Okay, so where did we get this OH group? If you observe your structure of your sugar group, deoxyribose, this is carbon 1, this is carbon 2, this is carbon 3. Okay, you can see here is a free OH group. You can see here, right? So this OH group and this phosphate group is what makes it the 5' and the 3' end of our DNA. Understood, students? So they are anti-parallel, and then they have these two ends. One is the 5' end, other one is the 3' end. The reason why we call 5' and 3' is because of what is present on the free end of our 5th carbon atom and the free end of our 3rd carbon atom, which is the phosphate and OH group, respectively. Now, if you see from above, like your water bottle, okay, we have our DNA, and I just look above. So what do I see is the diameter. So if I see the diameter of our DNA has the diameter of 20 degree angstroms. Okay, 20 degree angstroms, A here. Okay, 20 degree angstroms. Now, our DNA structure, they have something called as base complement, which is very, very important to understand its role. What is the base adenine always goes with thymine. Okay, always goes with thymine, and then we have cytosine, which always goes with guanine. Okay, sometimes you forget the words. Base complementation. Okay, see, I forgot. Okay, anyways, so adenine goes with thymine and cytosine goes with guanine. So this is called as base complementation. They complement each other. There is no possibility that adenine bonds with guanine or cytosine bonds with thymine. You have to remember this. Okay, so if you see one adenine of one strand, okay, if adenine is present in this strand, and the opposite strand will have what? It will have thymine. And they will have what kind of bond? They will have two hydrogen bonds between them. Double hydrogen bond will be present between adenine and thymine. Similarly, if we talk about the cytosine and the guanine, if cytosine is present in one strand and the guanine is present in the other, guanine will be present in the other strand, and between them there will be three hydrogen bonds. Okay, so you can see A double bond, T, again you can see C triple bond, G, or G triple bond C. So this is called as base complementation, and it's a very, very important property for our DNA. Why? Because so this complementation is what gives us a stacked nature for the DNA. What is a stacked nature? If I ask you to arrange coins in a tower, imagine some of you have mixed coins like 5 rupee, 2 rupee, 1 rupee, or even 50 paise. I don't know if you have seen that. Okay, of the coins. And one of the people, they're very smart and they're very lucky, and they have exact 2 rupees coin. So if they stack this 2 rupees coin, they make a very stable tower. And they make it in such a way that the diameter of the tower is equal, the distance between the coins are very, very equal. So it becomes a very stable tower. Similarly, this, because of this base complementation, now we have a very stacked nature of our DNA, which do not give gaps, much gaps. Like for example, if there is uh, let's take for example, if there is 0.34 nanometer of distance between one base pair and the other base pair, the same distance applies for all the bases that are present above them or below them. So that is what is called as the stacked nature of our DNA. So let us understand our complementation by this. If I have our 5' end and our 3' end of one strand, and we have the following base pairs such as A, G, C, T, 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 and then we have A, G, A. So if I close, oh, I can't hide it, I'm hiding on the screen. Anyways, if I ask you to write the opposite strand, it will be very easy for you. So you start with A goes with T, then G goes with C, and then C goes with G. Again, T, 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 A. Again A, again T, again C, again G. So this complementary nature is very, very nice to write also, and to even predict the other strand of the DNA. Now you have one advantage. If you have one single strand of the DNA, then you can easily predict the opposite strand of the DNA too, isn't it? So that is what complementation gives us, a benefit. Okay, and the next one, if you see the complementary DNAs which are twisted together like this, right? They're twisted together. So this twist, for example, from one turn, this is one turn, and this is the second turn, isn't it? So if you see the from one turn to other turn, what we see like this to this, so this distance is around 34 degree angstroms or 3.4 nanometers in length. Okay, what is the distance between each turn? Each turn is around 34 degree angstroms or 3.4 nanometer in length. Or you can see that if I start counting from one turn to another turn, what I can see is there are base pairs 1, 2. Okay, I'm not able to write 2. Give me a second. Okay, 1, 2, all the way till 10. So there are around 10 base pairs per turn. So if there are 10 base pairs per turn and each turn is around 34 degree angstroms, what should be the distance between two base pairs? Well, that means the distance between the 1 and 2, or 2 and 3, or 3 and 4 should be how much? You just divide the 10 between with the 34 or 3.4. What do we get? The distance between the two base pairs will be 3.4 degree angstroms or 0.34 nanometers. This is very, very important. Why? Because you students are quite in a hurry, and you just see 3.4 or 34 or 0.34, and you don't see the units that are mentioned at the end of it. So remember, if it is 34 degrees, it should be angstroms. So if it is 34 degree angstroms, we are talking about each turn. So if you're talking about 3.4 degree angstroms, we are talking about the distance between the two base pairs that are present in the in a DNA. And next one is when we talk about 0.34 nanometer, that is the same, the distance between the two base pairs that is present in the DNA. All right. So one second. Yeah. Sorry. So all good. You understand this? Okay. So let me go further and see what other characteristics that we have. So we understood that we have 34-degree angstroms and 3.4-degree angstroms as the distance between them. We understood the complementary nature. We understood the anti-parallelity of our DNA. And BDNA is a right-handed DNA. So why do I tell it is a right-handed? Because the turn that is there, it twists towards our right-hand side. Okay, it goes towards our right-hand side. It turns like a staircase. So that is why we call it as BDNA, as the right-handed helical DNA. Now, if you see our BDNA, the right-handed helical DNA, now one of the important characteristics that we can observe is between the two turns, we can come across these terms called as major groove and the minor groove. What is a major groove and the minor groove? So you can see that this portion of the DNA is called as a minor groove. You can see here clearly. So this is minor groove. Okay, this is minor groove. And this portion is called as the major groove. So what is this major groove and the minor groove? Minor groove is what you can see the small exposed portion. Major groove is something that you can see the larger exposed portion. So this shift happens, major groove, major groove, minor groove. And that keeps happening in our DNA. Okay. So this is about the double helical structure of the DNA that is proposed by Watson and Crick. Now, one of the major astonishing aspect of our Watson and Crick is, what they told. They understood the anti-parallelity, they understood the base complementation, and with this anti-parallel and base complementation, they also predicted something. Watson Crick was the first person to even tell that the DNA, if it replicates or if it makes copies, the way for it to make copies will always be a semi-conservative mode. But unfortunately, Watson and Crick will not be able to prove this experiment, prove this through experiment. That is why that statement remained and until it was proved. And when it was proved, they went back and were like, yes, Watson and Crick predicted this, and they really told about this. All right. So, chalo. So remember in the beginning, when we're talking about the double helical structure of DNA, I mentioned that the type of DNA that we are talking is the BDNA. Remember? So this is what I was talking to you guys, right? So there are other forms of DNA also. So what are the other forms of DNA that we have? Something called as aDNA and the zDNA. So we have other forms like aDNA and the zDNA. So usually they're extremely rare, and basically we can find them in laboratory. So let me do one thing. I will take the bDNA and I will try to dehydrate our bDNA. Dehydration is removal of water molecules in our bDNA structure. So when I remove the water molecules from our bDNA structure, now what is happening here is because of the excessive dehydration, now the base pairs come very close together. When the base pairs come very close together, that structure, which is a right-handed helical structure, is called as the aDNA, and the aDNAs can be only produced in labs. They are not found in any living organisms at all. And the left-handed helical DNA with a longer, see, we have 34-degree angstroms as the one curve, right? One turn. Whereas if you take with zDNA, the zDNA has a left-handed helix, but the base pairs are arranged in quite a distance. Okay, the distance between the base pairs and the distance between the turns is also higher in the zDNA. And the zDNA is very, very rare in nature, and usually it is found in some plant cells, and it is not functional also. So it has been cited in some of the plastids and the plant cells, whereas aDNA is strictly laboratory. And here, when we see the bDNA or aDNA or the zDNA, one thing is very common in all the three DNAs. So what is that? As mentioned by Watson and Crick, they say that if you take ladder as an example, and the two long sticks of a ladder, if you consider, okay, if you see these two sticks of a ladder, this is better, I think. Okay, this is only better. Okay, if you take the two sticks of a ladder, call these two sticks as the backbone of your DNA. And this backbone of the DNA is made up of your deoxyribose sugar. And to this backbone of the DNA, what supports is your phosphate group. So the backbone of the DNA is made up of a phosphate group and the sugar, whereas the base pairs project towards the inside. So how do they project towards the inside? So we know that in the first position of our sugar group, they occupy this position, the nitrogen group, and they project towards like this. And this guanine and this projected cytosine, they make these triple bonds or three hydrogen bonds, and that is what holds together. Understood? So we understood the backbone of the DNA, the composition of the backbone of the DNA, and how the base pairs project towards inside like the steps of the ladder. Now, the point is, what's the advantage of having a hydrogen bond in our double helical structure of the DNA? One important factor is our DNA is not an ideal structure. It is a constantly functioning structure, either in replication or transcription or translation, or for the expression of its characters. So that is why what happens is, if you see our DNA is quite like spring, it can absorb the shocks, it can twist, turn, and also it can get back to its normal shape and size. So this nature comes because of the hydrogen bonds elasticity. So the hydrogen bonds, whenever there is a torsional stress or whenever the DNA winding is happening for packing or whenever it is loosed for transcription or translation, all these places now you can twist turn without expecting the break in the DNA's helix. So that is why the hydrogen bonds gives very extremely high stability to the structure of our DNA. Understood? So here we complete the structure of our double helical DNA. So with this, Cherbox comes with the rule, and this is a very, very beautiful rule that explains the nature of the DNA, why the DNA is quite stacked. Okay, so it gives an explanation for everything. Why DNA is such a stable structure? Why not the distance between the base pairs is here and there? Okay, why it is always constant? So he said that always the purine to pyrimidine ratio in the DNA is always equal. That means we know what are the purines, right? The purines are adenine and guanine. If we add the total number of adenine and guanine, and if we add the total number of thymine and cytosine, they will always be equal. That is what the Cherbox mentioned. So this ratio. So if I write adenine plus guanine is equals to thymine plus cytosine, then I can also write it as adenine plus guanine can be divided by thymine plus cytosine is equals to 1. And this formula, students, is very, 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 very important. Why? You will understand when we solve the questions for this and the type of questions for this for sure. Okay, so this is a very, very important rule, and that gives the total explanation of why the DNA structure is such a beautiful architect without any mathematical mistakes or errors in arrangement of these base pairs. Understood? Sure. Next, can we predict the length of the DNA or the number of base pairs present in the DNA just by having one of the numericals? For example, let us say that our number of base pairs in the human DNA is 6.6 billion base pairs. That is 6.6 into 10 power 9. We have this much DNA in our diploid state. Okay, so what would be the length of the DNA? It's very simple. You just take the 6.6 billion into, you multiply it with 0.34 nanometer. So if you multiply with that, we get around 2.2 meters of our DNA is the length of human DNA. Okay, so similarly, you can take the E. coli DNA. The E. coli DNA is what? 1.36 millimeter. And then what do? You divide it. And then you get the number of base pairs. What do you get? 4 into 10 power 6 billion base pairs. Okay, sorry, 4 into 10 power 6 base pairs is what is the length of the E. coli DNA. That is 4 million base pairs, people. Understood? So we can understand the length of, we can calculate the length of the DNA, the distance of the DNA, or the number of the base pairs of the DNA having any of the two numericals. It's very, very simple. Okay, so now we have a very important topic, something called as the central dogma. Now, what is a central dogma? So before we head on, we have our beautiful new students in our class, and I would not be happy. Yeah, somebody is asking, can you explain the major and the minor groove? This is what I was expecting, students. So let's get back here. Okay. So let me go back before we go into the central dogma. So what is a major group and what is a minor group? You just have to observe this picture. So if you observe this picture, okay, can you see this part of exposed? And can you see the other part, that is this part of how much of the base pairs are exposed? If you take the proper, what do you tell, X-ray of our DNA, what you can see is, if you can see the X-ray, you can see a light band of this much, okay, of this much width, and another light band of this much width. Okay, for example, if I take my X-ray of the DNA, at one point I can see this much, and another point I can see something like this. Okay, something like this. So why does it happen? Just take a ribbon and twist it. So when you twist it, you can see that at one point you can see a small portion of the thing that is exposed to you, and on the other end you can see a large portion of this turn giving you more of its face, right? Like this. You can compare this with this purple. Okay, look at this pink color that we have. So just compare the amount of these bases that you can see with your eyes, isn't it? So this turn, which gives us more exposure, something like when you twist it, you can see that a large portion of it gives us a face, like a face. So this is called as a major groove. And if you can see a very small portion of it, that is called as a minor groove. You just have to do the simple experiment. Just take a ribbon and twist. And if you just hold it and see, you can see that there is a small band and there's a large band. So this turn that gives us a small band is called as a minor groove, and this turn that gives us a major turn, that is called as a major groove. Understood? So I hope you got it. Okay. So please confirm if you have understood it. Yeah, perfect. So can we go for the central dogma? Yes. So if you have further doubts, please let me know. So chalo, central dogma of life. What is the central dogma of life? Now, the reason why we have been studying about our DNA is because we were very curious to know what is that one particular thing that holds the characters, or what is that one thing that is responsible for the expression of the characteristics in our living organisms. So we understand what are characteristics in our Mendelian genetics, like yellow color, round color, wrinkle shape, or you can see that you can also talk about the red color eyes, brown color eyes, body, yellow color body. So anything that you can see and observe is called as a character, right? So this character, how does it come in place? Now, if you see our living beings, like just take an example of human. So if you take my example and if I take your example, what all I can observe in you? I can see your skin color, I can see your skin type, hair color, hair type, eye color, okay, and your eyesight, your intelligent quotient, your face shape, right? The ear position, okay, and the ear type, or the nose, the shape of your nose, okay, and then your gender, that is male or female, right? So many thousands and thousands and thousands of biology, lakhs of biological characteristics you have. Now, why did I mention lakhs of biological characteristics? You produce so many enzymes, you produce so many proteins, you do so many biological activities, you sense your environment, you sense heat, you grow, you just decay, you just die. So there are numerous, like thousands of functions are happening in your body. So for all this, we can call them as characteristics. So who is telling my cell to really function in such a way? Now, if I take a human body, our human body is made up of 3 trillion cells, around 3 trillion cells. That is what our body is made up of. Each of our cell has the capacity of independent life. They can live like a bacteria. They can just go away from our body, and they can live like a bacteria, like a unicellular organisms, without even bothering about what is happening to my body. It can be independent. But why our cells abide or follow the rules and just listens to everything irrespective of what your body is facing? You're healthy, your cells are with your You're not healthy, your cells are with you. They're always trying so hard to keep you alive, to keep you well, and to keep you functioning at all points. Not only the functioning, you're also adapting, evolving, and you're also reproducing, you're also producing generations. Like this is a very complex activity that is happening as a living organism. And how these activities gets dictated, like your country, you have governments, you have offices, and you have so many different levels where the information gets passed and where the people and the society live together. So similarly, even in our body has its own governance. And how does it happen? We have cells. Cells have nucleus. Nucleus have chromosomes, and the chromosomes are made up of DNA. And we know that the DNA is the genetic material. Now, what happens is the DNA is a very raw information that we have. And out of 100% of the DNA that we have, not 100% of the DNA is giving us the messages, or it is useful, I can tell, or it is participating in, controlling the life. Only less than 2% of the DNA that is present in our body is what is functional. That means it is what is controlling all this that you see, all this that you feel, all this that you read in biology is what is just 2% of our DNA. So this functional DNA that we have is called as the genes or the factors, what Mendel mentioned. So these genes are the factors, so which are in the rawest form of the information. Now, this which is in the nucleus have to be sent as a message to our cellular components or cellular organs to do a specific function. And how does it do it? So this DNA, the messages that are hidden in these genes are converted into a readable message through a process of transcription. Okay, we'll talk about transcription, where the message of the DNA is decoded into a readable form for our cellular components. So that is called as RNA, or the ribonucleic acid. So this RNA comes out of the nucleus in the form of mRNA or messenger RNA, which carries this genetic message or to our cellular organelles. So once it goes to the cellular organelles, our cellular organelles use this message and read it to construct proteins. So this proteins is what behaves, and it's what is responsible for the expression of characteristics. So what are you talking about? We have carbohydrates, we have lipids, we have vitamins, we have minerals, we have so many things. And you're telling us proteins are responsible for all this? Yes. 99% of the biological functions that you can see, feel, hear, whatever it is, it's all because of proteins, and proteins control them. Understood? So this chain of events that we have, that is from the DNA to RNA and RNA to protein, is called as a central dogma of life. That is called the flow of information of life, or the flow of life, that we can simply call it as. So this is about our central dogma. And to understand the central dogma, we have to understand how the DNA is present, how it is packed, and how does it get replicated, how does it get expressed in the coming classes. So now we are running out of the time. Okay, so I conclude this class here, hoping that you understood what we spoke today. And in the tomorrow's class, we will continue on from our central dogma of life. We'll see how the packing has been done in prokaryotes, and then we'll see the rest of the interesting topics. Okay. If you have any doubts, you can leave a message now. If you do not have any doubts, thank you so much for attending today's class. It was exciting to see so many people here in this classroom. I'll see you tomorrow. All right. Thank you, students. I'll be here. You can just leave a message if you have any doubts.